All right, now that we have finished our whirlwind tour of infectious agents or microorganisms, now we're going to talk about the principles of infection. So the learning objectives for this, I want you to define the terms infection, endemic, epidemic, and pandemic. Not our favorite word, right? 5 modes of transmission of infectious diseases. The primary factors affecting host susceptibility, that's pretty important. I want you to name the factors that increased microbial virulence, and we'll talk about what that is. Explain 3 reasons why an infection might occur, might not occur after the pathogens enter the body. Being able to list the signs and symptoms of infection, and listing methods of diagnosis that are used to detect infection in the medical world. So an infection occurs when a microbe or parasite is able to reproduce in or on the body's tissues. So infectious diseases may occur sporadically in a single individual. They can be localized in groups. They can be, an endemic is a continuous transmission within a population. So we can think of sort of colds and flu as being endemic. They're always around. We always have to work on not getting them. And it's just always going on. An epidemic, so certain infections that occur outside their normal geographical ranges or in higher than expected numbers, these are referred to as epidemics. So let's just say in this particular geographical area, a certain infection is endemic to that area. All of a sudden it spreads to a new geographical area, then it becomes an epidemic. When transmission has occurred on most continents of the world, it's known as a pandemic. So really knowing the modes of transmission of microorganisms and the methods of controlling it is essential for prevention and control of infection within the community. So this has been, unfortunately, a major focus for everyone in the last few years. And really in healthcare, it's always been a focus. So we'll talk more about it. So a chain of events occurs in the transmission of infectious and infecting organism from one person or organism to another. So you have an infectious agent, whatever it is, it's the microbe causing the infection. Then you need a reservoir. So the reservoir is a source of infection. It may be a person with an obvious active infection in the acute stage, or it could be a person who's asymptomatic and shows no clinical signs or symptoms. So even though you don't have symptoms, you can still be a reservoir of disease. So if you isolate the reservoir of disease, you can break the chain right there. So that's something to think about. If you have someone who doesn't have any clinical signs, they might be early in the incubation stage of infection. They might be a carrier of the organism where they're not going to ever develop the infection. So people who have subclinical forms of the infection that's really mild and has a little or no manifestations, they're considered carriers of the disease. So they're still a reservoir. So once you have a reservoir, well, the reservoir, besides being a person, it could also be an animal, contaminated water, soil, food, or equipment, depending on the method of transmission, the mode of transmission of the agent. So there are lots of different modes of transmission, which we'll talk about. But you need a portal of exit, how the agent leaves the reservoir. So if it's an airborne illness, a sneeze, it can be the portal of exit. It goes out through your nose, or just talking, little droplets if it's droplet transmission. So you need some kind of portal of exit from the reservoir. The mode of transmission is how the agent reaches the new susceptible host. So it can be through air or water. It can be by direct contact. It can be through food. So direct contact with no intermediary, so like you're touching an infectious lesion, or sexual intercourse is direct contact. Microbes can be in blood or body secretions or a lesion. So not all microorganisms can cross blood-brain barrier or placental barriers, but some microbes that can cross the placenta can really cause problems with fetal development. So there are some examples in the book where this is a problem. Indirect contact involves an intermediary like contaminated hand or food, or a fomite, which is an inanimate object like instruments or bed linen that carries microorganisms. Or like, say you sneeze into a Kleenex, you set the Kleenex down, somebody with an ungloved hand, or a gloved hand even, picks up the Kleenex, throws it in the trash, and then wipes their eye with it, or scratches their face or something like that. That is indirect contact, but you can still get that. So sometimes there are food or other things in the environment that can participate in indirect contact. So direct contact, no intermediary, you're touching the part that is infected, or infected blood or body secretions. Indirect contact involves intermediate object or an organism. It could be an inanimate object like a dirty surface, or non-sterile instruments, or a towel, something like that. Droplet transmission is respiratory or salivary secretions that are expelled from the infected individual. So droplet transmission is can happen through just talking, just normal talking. As we're talking, there are little microscopic droplets, and if you're infected with a virus, you can pass that on to someone just through their respiratory or salivary secretions. If you yell, or if you're breathing hard, or if you sneeze or cough, it propels the droplets even farther. But just normal talking can be involved in droplet transmission. So it can be oral or respiratory secretions. And they can be inhaled directly by another person close by, or fall on nearby objects to be transmitted indirectly. Aerosol transmission involves small particles from the respiratory tract. So they're suspended in air, and they can travel farther than droplets because they're not as heavy. They're not going to fall as quickly. Vector-borne is when an insect or an animal serves as an intermediary host, like a malaria. You get it from infected mosquitoes. So lots of factors can decrease the host resistance. Age, if you're really young or really old, that can decrease your host resistance. Pregnancy, because the pregnant woman's body is working really hard to build this new human being, and it can decrease their resistance. Genetic susceptibility, nothing to be done about that. Immunodeficiency, because your immune system's not working as well. Malnutrition, probably because your immune system's not working as well. Chronic disease, same thing. Severe physical or emotional stress, inflammation or trauma, or impaired inflammatory responses. And in the next chapter, we're going to talk about inflammation specifically and the processes that go on in the body. But an inflammatory response can be a symptom of an infection, or it can be something else. It doesn't have to be involved with infection. But impaired inflammatory responses can affect some of our lines of defense, protecting us from pathogens. So we talk about virulence and pathogenicity. Pathogenicity is the capability of a microbe to cause disease. So that just means that it's a pathogen. It's something that could potentially cause disease. Virulence is the degree of pathogenicity. So a bacteria or a microorganism that has more invasive qualities, like it's has higher motility, or it has those exotoxins, those enzymes that bacteria create, can make it more pathogenic. Toxins, adherence to tissue by pili, fimbriae, or specific receptor sites. So a lot of viruses will bind to specific receptor sites on specific cells, and that makes them more pathogenic. And the ability to avoid host defenses, that makes a microorganism more pathogenic as well. So newly emerging diseases can be an issue. Antigenically different forms of common infections, such as flu, influenza, or COVID. So a new mutation can better evade the host defenses. It can better get by our barriers against that. So anytime a virus mutates, it makes it potentially more pathogenic. If it spreads beyond normal endemic areas, you have people that don't have haven't built up immunity to it. So that was one of the things that got the whole world with COVID-19, is none of us had immunity to it because it was a newly emerging disease. And unfortunately, it killed you on the first time. So you, for a lot of people, so you didn't have a chance to develop normal immunity to it. Super infections, they're considered multi-drug resistant forms of existing diseases like tuberculosis and Staphylococcus aureus. Those are bad because we can't fight them with the tools that we have. So that can be really dangerous and interesting. So my little bacteria cartoon there. You could enjoy that on your own time. So this is one of the lovely boxes in the book, saying what our resistance is, and then what can cause increased microbial virulence. So having intact skin and mucous membranes, that's a line of defense in our resistance. So exotoxins and endotoxins from bacteria can break that down. Body secretions like stomach acid and tears, they have chemicals in them that can help break down bacteria before they get to the parts of our body that are vulnerable. Microorganisms can produce destructive enzymes that can break those down. Non-specific phagocytosis. So we have cells going around, and we'll talk about this more in the immune system chapter. They're just going around, cleaning up the place, getting rid of potentially dangerous microorganisms. Microorganisms can form spores, and so maybe they're not recognized by non-specific phagocytosis. Having an effective inflammatory response can be a good way to increase your host resistance, and we'll talk about that in the next chapter. If there are a large number of organisms enter into the body at the same time, it can overwhelm our inflammatory response. Absence of disease, that's a good that's a good way to have resistance, just like be overall generally healthy. An effective immune system interferon production fights against viruses. Bacterial capsule and pili kind of are another factor, other factors in microbial virulence, because they can stick to cells better. So it's just something to think about. So talking about our body defenses, we're going to talk really super specifically about this in chapter 7, but just a review of these. Our first line of defense is non-specific, and it's a mechanical barrier. It's unbroken skin and mucous membranes, and our secretions in tears and gastric juices, which have things that break down microorganisms. So if you're keeping your skin intact and mucous membranes intact, that's a really good defense, our first line of defense. If things get past the first line of defense, which they do, the second line of defense is also non-specific. It's anything that comes in. So we have those non-specific phagocytes that are going around just looking for things and cleaning it up. Inflammation triggers a chemical cascade that also calls more phagocytes to the area. And so that's non-specific, but it can help in our second line of defense. Our third line of defense is specific defense, and that's what our immune system is responsible for. So our immune system produces specific antibodies or cell-mediated immunity that is going to be a specific immune response to a specific pathogen. So if the pathogen overcomes all your defenses, then it results in injury or disease. So that was one of the learning objectives, is how can a pathogen cause injury or disease? It goes through all the levels of your defense and it breaks it down. So we have those specific body defenses to kind of protect us. So with infection, there is an incubation period, which is basically the time between the entry of the organism into the body and the appearance of the critical signs of disease. So the incubation period can vary considerably with different organisms. So an organism can have an incubation period of a day or 2 days or 2 weeks or whatever. So part of the study of disease and the physiology of infection is figuring out what the incubation period is for individual organisms. So the prodromal period, and we talked about this in the first chapter in terms of terminology, it's where you just don't feel right. It's non-specific. You don't have specific symptoms necessarily, maybe fatigue, loss of appetite, maybe a headache, kind of feel like you're coming down with something. The prodromal period can be more evident in some infections than others. The acute period is the period in which the infectious disease develops fully and you have clinical signs and symptoms and you know you're sick. So infections can take on some different patterns defined by their characteristics or locations. So local infections are where an organism enters the body and remains confined to a specific location. Focal infections, the pathogen spreads from a local infection to other tissues. Systemic infections, the infection spreads to several sites and tissue fluids. That's particularly dangerous with systemic infections because tissue fluids are traveling through the circulatory system. And so now you've got a distribution system. So septicemia can kill you. Septicemia, so anything with emia means blood. Septicemia means you have a blood infection. It's caused by multiplication of pathogenic organisms in the blood and is the cause of sepsis. So sepsis is a toxic inflammatory condition arising from the spread of microbes. Septicemia is really dangerous. Bacteremia is, I mean, if you break the word down, it's presence of bacteria in the blood. Toxemia is presence of toxins in the blood. And viremia is presence of viruses in the blood. So you can have mixed infections where several infectious agents are concurrently establishing themselves at the same site. Acute infections appear rapidly with severe symptoms but are short-lived. Chronic infections have less severe symptoms than acute, but they persist for a long time. So just like our acute and chronic that we've talked about with regard to other things, that's the same definition. So secondary infections, well, primary infections are the initial or first-time exposure or infection. Secondary infections follow a primary infection and are caused by a microbe other than the one causing the primary infection. Opportunistic pathogens are often the cause of secondary infections. So this is like you get a cold, your immune system's busy, and then you get a bacterial infection following the cold. That's your secondary infection. Because your immune system is, the opportunistic pathogens go, hey, she's busy, let's take advantage of that. So a subclinical infection doesn't cause apparent signs or symptoms, but it can become chronic and persist over long periods of time because it doesn't cause those symptoms. So think about the last time you had a cold and compare the prodromal period with the acute period. I think we've all been through this, and so we all have a first person relationship to it, and we can understand what's going on. So the signs and symptoms of infections can be local or systemic. So local signs of infection include inflammation, pain, swelling, redness, and warmth. If it's bacterial, it's a bacterial infection, you could have a purulent exudate, which means pus. It's exuding pus, purulent exudate. If it's viral, it's usually a serous or clear exudate. So those are local signs of infection. Systemic signs: fever, fatigue and weakness, headache, nausea, generally what we refer to as being sick. So local signs versus systemic signs. You should know the difference between those. So with bacterial infection, you also can see systemic signs in the blood count, the different cells that are present in the bloodstream. Leukocytosis is where you have an increased white blood cell count, and frequently with bacterial infections, you have that increased white blood cell count. So when they do basic blood tests, they usually do a complete blood count, and they compare how many white blood cells you have to the normal level. So knowing the exudate type and the systemic signs is important for helping to diagnose the illness. So things that are used for diagnosis of infections: culture and staining techniques. Usually the bacteria can be cultured pretty easily, and they can be stained to know if they're gram positive or gram negative. You use specific clinical specimens like nose swabs, whatever. They're trying to determine if those particular microorganisms, what they are and how to kill them, basically. So blood tests, variations in number of leukocytes, white blood cells, can determine whether it's a bacterial or viral infection. So leukocytosis is elevated white blood cell count. That indicates a bacterial infection. Leukopenia is depressed white blood cell count, and that indicates a viral infection. A differential count is where they count the different types of white blood cells. So changes of the distribution in leukocytes can be different depending on the organism that's infecting. So neutrophils tend to increase with acute infections, but lymphocytes and monocytes increase with chronic infections. C-reactive protein and erythrocyte sedimentation rate, or ESR, are usually elevated and are a general indicator of inflammation. So blood tests are also useful for detecting antibodies and confirming a diagnosis, particularly in a case of viral infection. So when you take your little home COVID test, you're testing for viruses. you're testing for the viral antigen, basically. And so those rapid tests can be based on metabolic characteristics and on serologic characteristics of organisms. So there are lots of different rapid tests like strep tests, COVID tests, flu tests. There are different rapid tests that you can take now. And usually they do that when you have an active infection. So immunological testing of body fluids, antigen identification is identifying that you have that virus or bacterium in your system. The antibody titer is telling whether you have antibodies that react to that antigen. So when I was talking about in another lecture, getting the varicella zoster titer to see if you have antibodies to chickenpox, that is the immunological testing of body fluids. That's the antibody titer.